Sacramento State, and I'm pleased to welcome you to our talk tonight. This is presented by the College of Natural Sciences and Mathematics and the Sacramento State Center for STEM Excellence. And STEM stands for Science, Technology, Engineering, and Math. The center works with area schools, colleges, universities, and employers to create a seamless STEM pipeline for a scientific and technical workforce. This is the fourth presentation of the year in our series of public lectures on regenerative medicine, featuring prominent scientists in the field of stem cell research. The lecture series is funded by the California Institute for Regenerative Medicine and supports our master's program in stem cell research in the Department of Biological Sciences. We are very excited to announce that our second cohort of stem cell master's students will graduate this coming May. Students in our third cohort have already started conducting research with our partner institutions in this program, including the UC Davis Stem Cell Program, and we are currently recruiting for next year's class. As part of disease teams, our master's students work with scientists and physicians on cures and treatments for disorders including neurodegenerative disease, spinal cord injury, HIV infection, blood disorders, and burns. Sacramento State graduated its first class of master's students in stem cell research last May. It now gives me great pleasure to introduce this evening's speaker, Dr. Fernando Fierro, a stem cell research scientist at the Institute for Regenerative Cures in Sacramento. Dr. Fierro received his BS in molecular biology from the University of Chile in Santiago in 2003, and his PhD in cell biology from the Technical University in Dresden, Germany in 2008. Dr. Fierro has been a postdoctoral fellow in the laboratory of Dr. Jan Nolta at UC Davis since 2008 and is the author of over a dozen research papers and articles. Please join me in welcoming Dr. Fernando Fierro.
Hello. Thank you very much for coming, and thank you very much for the invitation to STEM. This is a great honor and a privilege to be here. I will be presenting for about two themes. One is MSCs in translational research, so it's very applied. It's about stem cells and how we can really use stem cells to treat diseases or malignancies that we haven't found a cure for them right now. And the other part of the talk will be focused on microRNAs and MSCs. And it's really from very basic questions that we are approaching it. But of course, you will notice that it cross-links all the time between the basic research and the translational research that we do in the lab. And I think this is something that happens all the time in all disciplines in biology. We do something maybe just because we are curious and we want to know how it works, but it ends up almost always having some implication on how we can now treat patients now that we have this new knowledge, and that can improve either the therapeutic approach or the diagnostic tool, develop new diagnosis tools, and so on. Okay, so for the first half of this talk or so, I will be talking mostly about what are mesenchymal stem cells and why are they so popular and so on. So this starts, again, as I just mentioned, from a very basic biology problem. And it's how did it happen in evolution that small animals like flatworms, you can cut them in almost 300 pieces, and each of these pieces is able to regenerate the entire animal again, right? And this is also true for fishes. You can cut the fin, and the fin regrows, or some organs like the heart, or as you well will remember, the amphibians like salamanders and newts, you can amputate the tail, and the tail just grows perfectly back to what it was before. And the same if you actually cut off a whole limb. The leg knows exactly how, there is some sort of memory there contained in the amputated site that allows them to regrow and form exactly what was there before. That's what we call regeneration, right? So having a tissue damaged, we are now trying to come back to exactly what we had there before. And to some extent, we know that that's still true for us. For example, if we break a bone, there are good chances that we're going to fix that bone by ourselves, right? And this is because precisely we are assuming that those cells that are responsible for the regeneration process are actually conserved among all these animals. It's what it's called a blastema in this organism. This is a layer of quite undifferentiated cells from mesodermal origin in evolution, in development, don't worry about that. But it's basically saying that we have, even as humans, to some extent, some regeneration potential, right? It's not as perfect as in salamanders. If we cut ourselves a whole finger, the finger will not regrow as it was before. Now, of course, we want to understand why is it that we lost that potential. Why did lower animals during evolution could do that, and what did we do that we now lost that potential? And what I will show you is how much mesenchymal stem cells are probably a promise to achieve a lot of this regeneration again. I call them mesenchymal stem cells really because I've been just working with them for the last 10 years with that name. But there are so many other ways to call the same cells, really. Other people call them simply bone marrow stromal cells or multipotent stromal cells. The good thing is that any of these different names always end up with the same abbreviation, MSC. So we agree that they are MSCs. And this is just a brief video that I did now quite a few years ago, where actually the MSCs are the cells that are in the bottom of this image, a confocal image. It's a 3D reconstruction, yeah, okay? So these are hematopoietic stem cells, cells that give rise to the blood. And the reason why I prefer to show you this picture is because even though the term MSC is rather new, has been just being used for the last 10 or 15 years, the cells that are supporting the hematopoietic stem cells are well known for many more years, and it's really the same cell type. The only difference is that by 2003 or so, we discovered that these supporting cells, or niche cells, actually have a differentiation potential. So they have a capacity to become other cell types. Not only the niche cell itself, but they can actually differentiate into osteoblasts, so bone cells, or adipocytes, fat cells, and chondrocytes, cartilage, and many other cell types. All still limited to what we call the mesenchymal lineage, mesodermal in development. So it's not very popular, the idea, even though there are researchers working on it, on, for example, trying to differentiate these cells into neurons. And in this case, we are saying that mesenchymal stem cells are probably not the very first alternative to think on, for example, you know, all this discussion that has been about using or not embryonic stem cells, where clearly embryonic stem cells have a differentiation potential, this capacity to become other cell types much larger than what mesenchymal stem cells can do. The mesenchymal is limited to mesodermal lineage in terms of their differentiation. But the real reason why these cells have become so popular are the listed three here. They are very simple to isolate, they have a great expansion potential, and they have low immunogenicity. And you will see that these three facts have made them extremely easy to obtain and to put back in a patient in a very safe manner. And that has been the first ground that has now made us think about, well, for what can we actually use these cells? The first fact, you have to think that every time we're thinking on a therapy, we care only about two things, really. It has to be safe, and it has to have some efficacy. And the mesenchymal stem cells are the only cells that have demonstrated by far the most amazing safety that you can imagine, actually much safer than to try to transplant, for example, skin from one person to another, because you normally would reject, of course, the cells that are not your own cells. And that's not the case for mesenchymal stem cells, as I will show you later. How we isolate these cells? Well, I will keep it pretty simple, and I will tell you that we do it from bone marrow. So any of our large bones have marrow inside, and from any of those you could obtain mesenchymal stem cells. Now, if I want to be more precise, mesenchymal stem cells are really present in any tissue. Any vascularized tissue has high amounts of mesenchymal stem cells. And that makes, for example, now a very common source to isolate these cells has been adipose tissue, right? Because a patient that is, for example, undergoing a liposuction is taking all this material out of his body. Normally it's being wasted. Now we are finding it absolutely full of stem cells, and we are saving all that material because it's extremely rich in cells that, as I will show you later, have an amazing regenerative potential. But I want to focus for today on bone marrow mesenchymal stem cells, basically because these are the best characterized cell type, even though we think that they are really, really common features to all these other tissues from where we can isolate the cells. And because all the data that I will show you, the results of our own research has been done on cells isolated from bone marrow. So it will keep it much easy and simple. Okay, well, so you basically do, you have to do an aspirate. Typically it's aspirated from the hip, from the upper iliac crest, because that's a large volume. So you can take up, let's say, 100 milliliters of bone marrow and you will be okay. It actually causes some pain to the donor, but many patients have to go under that aspiration because, for example, they want to look how their bone marrow is doing. Anyone who has a blood problem, they will have to go under this intervention just to test how their bone marrow is, in what conditions it is. So that material that normally would have been used just for analysis, now we are using it to expand these cells. There is a very simple isolation protocol, which really the only thing that does is to take off the red blood cells, right? Because the marrow, as you know, is very red. So we take those cells out, and we want to stay with only the mononuclear cells. And when you plate that in plastic flasks, something amazing happens. Almost no other cell attaches, right? The cells normally of the bone marrow are floating in that bone marrow, you could say, except for this cell type. You see that there's a single cell here that is kind of a long thing. Here are a lot of actually red blood cells, so you can tell that the purity is not 100% at the moment of having done this earlier step. And then you have a lot of other cells that are in the bone marrow, and they will not attach. So after these 2 days, we do a very simple process, which is to wash that flask and remove all the cells that did not attach. After 5 days, you will see that these cells have actually expanded quite a lot. There are more cells, and as I mentioned you before, these cells are supporting cell for other cell types. So these other cell types actually are trying desperately to attach to these cells. They're not going to be attaching to plastic. They don't have that potential, but they definitely have the potential to interact in a cell contact with the mesenchymal stem cells, with the MSCs. And but if we keep washing this culture, you will see that after a few days more, 10 days in this case, we have reached a high level of purity of culture. So now these cells are basically in the absence of any of these round, shiny cells, which are typically hematopoietic cells that will have something to do with the blood. And we will end up with these other cell types that, of course, resemble a lot fibroblasts from their morphology. Okay, so as I mentioned to you before, the key point of this step is that we are isolating the cells from a very simple principle, is the fact that the cells can attach to plastic. And what have a huge implication for their therapeutic potential is the fact that we can actually grow them so much. For example, hematopoietic stem cells, as I mentioned before, blood stem cells, are of course used in the clinic for more than 60 years now. Since the late 50s, we have been doing bone marrow transplant. So stem cell therapies are an old story. But those cells, you cannot take them out from a patient and make more to treat more than one patient. Actually, it's very difficult to find that the cells that you are taking from a certain donor will match whoever is needing those cells. Normally, those cells will be rejected. So even though it's a very common practice, it's very limited to how many people can really be treated with hematopoietic stem cells. That's quite different for mesenchymal stem cells, right? We are taking out, let's say, 100 milliliters from the bone marrow aspirate. There are approximately 30 million cells in each milliliter, and the number of MSCs there is very variable, as you can imagine and probably guess. When we are children, we have a lot more MSCs in our bone marrow than when we are old. And the more we age, the less of these cells we still have in our body. That makes, among others, how children can repair their bones and their cartilage fractures like that so easily. And as we get older, we have less and less of that potential to regenerate mesenchymal tissues. So these cells, anyway, in average, are around 1 in 30,000 cells, so it's a very rare cell. And you would therefore take from 100 milliliters only 100,000 cells. Now, 100,000 cells, you can probably imagine that actually because, well, that's extremely little. I don't know how to explain it really. It looks like if you would have them in a tube, you would probably see a little ball there. It's something that you can see already with your naked eye, 100,000 cells, but it's extremely little. So if you're thinking on, for example, you have a bone fracture, you have a piece of bone, a non-union fracture, let's say, where you have a whole inch of bone missing, you're not going to repair that with 100,000 cells. That's not making anything, actually, right? So mesenchymal stem cells, again, the great difference is that we actually, only after 10 days, now have expanded these cells logarithmically, right? And instead of talking about 100,000 cells, we're talking about 100 million cells. And we don't need to stop after 10 days. We can culture the cells for a month or 2 months and whatever is necessary and obtain so many cells that will now be significant in order to repair tissues. So in other words, we are saying that the cells that would normally be repairing your tissue are just not able to do it sometimes because they're just not enough. So all we are really doing is take the cells out of the donor, multiply them in number, and put them back to the same person or a different person, as I'm going to show you now. Because mesenchymal stem cells, in contrast to, as I mentioned at the beginning of this presentation, are very well tolerated from one donor to a completely unrelated person. It's what we call allogenic transplants. You don't need to be, they don't need to take the cells from your own body to give them for you. It's actually very rare that that's how they have been used in the clinic. It's much more common that they take the cells from any donor, unidentified. We multiply these cells many times. We characterize them very well. They are all coming from the same batch, from the same person eventually. And now these cells will be sufficient to treat at least 10 patients for very different things. And that's what I will show you later for what we can use the cells, right? So a very important step is a critical step for any of these cell therapies is the fact that these cells are not being rejected when you use them to treat whatever in another person. Actually, if they would be directly injected in your blood, they're definitely not going to do anything bad at that point. It's very hard to tell what they would really do. Probably they will die after some time and you won't notice anything. But most important, your body will not have created an immune rejection against it. So you don't get an inflammation or things like that, as you normally would do, of course, if you are receiving the cells from an unrelated donor. Actually, the reason why I'm now here, the reason I don't want to go too much into detail why these cells are not being rejected. But another feature, which I think is very important in terms of why the cells have been used so much in the clinic for this application, is because precisely they don't only silence themselves, but actually they silence your entire immune system, right? That's what we call immune suppressor cells. As you can see here, mesenchymal stem cells can be targeting all type of lymphocytes and NK cells, monocyte and antigen-presenting cells, and so on. So mesenchymal stem cells actually have been used in way more cases. I'm saying by now it has to be somewhere between 4,000 and 6,000 patients to treat autoimmune diseases. It has nothing to do with the fact that these cells are stem cells and can differentiate into bone or fat cells. It's not that a characteristic of the cell that has made them so popular in the clinic. It's simply the fact that they are great immune suppressors. So for example, for graft versus host disease, I'm going to show you here, I will show you that in a minute, on how they can be used. It's much more common to be using the cells to simply downregulate the immune response that the body is having against something else. And this is the reason why mesenchymal stem cells actually have been mostly used in the clinic so far. Okay, so just very general, I want to say one more time the 3 reasons why mesenchymal stem cells are so popular. One is because they're very simple to isolate. We don't need to have antibodies or any technology like that. It's really just the fraction that can adhere to plastic. They have a great potential to be expanded. We can multiply the cell number with a very simple media, which makes them also, from the costs point of view, very low compared to the expansion of any other cell type. And they present this very low immunogenicity, right? And as I told you, well, great, the cells are safe, we can make a lot of them, but are they good for something? That's what I want to talk to you about today. So the MSC-based therapies are of two types, and I think I kind of give you a hint of that already. From one point of view, the cells can really differentiate, right? And that's what I would say, for example, you have a broken bone, imagine it like a hole in a wall. The cells are really coming to replace fill that hole, right? They will become like the bricks to really close that wall, and they're fully differentiating into whatever is missing in that cell type, in that tissue. And the most common application for this has been bone and tendon damage, and although many patients have been treated with it for these reasons, I would say that it has been much more popular in terms of how many patients have been treated. I shouldn't say patients, maybe. It's been in the field of horses, actually. So horse races, for example, it's very common they have a broken leg or something like that. Normally, that's the end of the horse. It's not going to come back to race. And now the mesenchymal stem cells have made a huge difference on treating them to bring them really back, regenerate the tissue completely. Remember that the opposite of regenerating is scarring, right? To make a scar is not a functional tissue. It's just collagen crossed over. It closes the wound. You are not bleeding, but that's not really the bone or the cartilage that was there before. So we are trying to avoid that part and making the cells really become whatever is really missing. But as I mentioned earlier, the most common application of mesenchymal stem cells had to do with something that they are secreting. It has not to do with the cells differentiating directly into a specific cell type. So in this case, we call them trophic mediators, and as I mentioned also, a very common application for this has been graft-versus-host disease or Crohn's disease, fistulas, and all these have been problems where there is certain damage, but the mesenchymal stem cells are not coming to close the wound directly, differentiating in whatever is missing in that hole. They are rather sitting there and just recruiting the right cell type to the site of the wound. So they are, first of all, of course, avoiding the inflammation, stopping the immune system of reacting, and they're calling, for example, for angiogenesis. There is no tissue that can repair without good blood vessels formed around it. So a very important step is to just recruit blood vessels around the wound. And this is for what mesenchymal stem cells actually have been mostly used. And I would show you actually an application in this regard. Yeah? This is, for example, project where we've been working where it's about patients that have typically diabetes, and they have ulcers in their extremities, in their legs, that don't close. So they are chronic wounds. They are wounds that stay open sometimes for years. Sometimes they get, of course, much worse. Eventually, it could develop into a gangrene, and the leg has to be amputated or so on. What is actually now used in clinic is, of course, a patch, just a scaffold. It's basically like a sponge that tries to somehow mimic the skin, put it on the hole to at least close that wound, and that's the best we can do so far to treat that chronic wound. Yeah? That's what I would call an acellular construct, where we are, of course, hoping that it will eventually become vascularized. The blood vessels will enter skin cells, keratinocytes, and epidermal cells will migrate towards that scaffold and will eventually close the wound. Yeah? But that's our best hope. Actually, it doesn't work that well. And typically, those bandages have to be replaced many times in the life of a person that has that type of ulcers. Our approach is now to what we call bioactivate the scaffold. So we fill the scaffold with mesenchymal stem cells, and we don't see any differentiation of the mesenchymal stem cells into skin cell types, so they are not really becoming keratinocytes or so on, but they are definitely contributing, particularly by recruiting new blood vessels around the site of the wound. Yeah? So we're going to put mesenchymal stem cells into these sponges, and we will see how those get repaired. This is our model. This is a newt mouse, and newt mice don't have a thymus, so they don't have T cells. And then we can study human cells in the mouse without being rejected, right? So that's why we use this type of mice that don't have fur. And we do a model where we have to do an excision. We take off a piece of skin, and this is now the sponge, the scaffold that we are putting back. This is actually a scaffold that is used in the clinic normally to treat this type of wounds. Yeah, it's called a Integra matrix. It's basically collagen. I will show you later that in more detail. But we put it with or without MSCs. Yeah, and we close the suture, and then we wait for 2 weeks, and we look later on how blood vessels have been able to form in that tissue. And this is how it looks in a normal scaffold that didn't have MSCs, and this is with the MSCs. Again, the MSCs are not even differentiating into endothelial cells, so they are not really being the red part that you see here. They are simply recruiting, circulating blood vessels to come into this tissue. Yeah? And that's most of the part that they're doing. This can be quantified by a digital segmentation software, things that don't matter for now. And this is a project where we have been working with 3 groups. One is Dr. Egana in Germany. Dr. Nolta is my principal investigator with whom I work here in the Institute for Regenerative Cures. And the dermatologist of the team is Dr. Rivka Iserov, also at UC Davis. And together, the 3 groups are trying to develop this cures. Because the goal is not only to put the cells there, but actually to find ways on how to optimize the cells that are going to plate it there to make this really a clinical application and not only like a nice animal model. Another collaborator we have in this team is Dr. Peavy, who's actually here, Tom. And he did very beautiful confocal images. This is a movie which basically, I think I'll show you the movie one more time. But the critical step is that this is not really happening in time. This is really a 3-dimensional imaging of the scaffold with the mesenchymal stem cells in it. So all what each of these images represents is a movement of about half a micrometer, higher and higher and higher. It's to have an idea of the true 3-dimensional distribution of the MSCs inside that scaffold. Yeah? So that's what you see. This is, for example, now the bottom of the scaffold, if I'm right. So it ended up with almost just green. The green part is just the autofluorescence. Let me show it to you again. This is, by the way, the scaffold alone, without mesenchymal stem cells as seen under an electron microscope. Okay, very nice, but nothing of that is alive. This is now with mesenchymal stem cells. The mesenchymal stem cells were stained. Their cytoskeleton, actin, was stained with a dye called phalloidine, and the nucleus are looking white. And the green part is the scaffold itself. So it looks like moving, but what is really moving, you imagine it is as we would be moving the camera along the scaffold, right? And it shows basically on how well the cells distribute in it. This is something that Dr. Peavy could quantify and observe of, well, if we put the cells on top of this scaffold, are they really going to stay all on top, or are they going to all fall through and go to the bottom of the scaffold? You can see that they actually distribute quite homogeneously. Most of them are maybe within the first half, and then there are less and less by the end of the scaffold. Okay, and this is the type of characterization we want to do. We also use many other type of scaffolds to see, well, really, can they distribute equally well in any of them? Definitely not, right? If the scaffold, for example, would have a too small pore size, it wouldn't allow the cells to go through. You would end up with all the cells on just one side. If the pore size would be too big, or it would be, all of these scaffolds are meant to be biodegradable eventually so that they can really be absorbed by your body. If they would be too degradable, they would probably break up without the cells in them, so that doesn't help either. Now I want to show you a few examples of experiments where we really try to characterize the functionality of the scaffold. Yeah? And one of them is, of course, the secretion of factors that recruit other cell types. This is ELISA. It's a technique to measure the abundance of a protein that has been secreted by the cells. In this case, we're looking for a chemokine called stromal-derived factor 1, or CXCL12, which is very well known to attract endothelial progenitor cells, so the cells that are responsible to form new blood vessels. And you can see that in the scaffold, as this go along, they secrete more and more of this scaffold. You can detect more and more of this. These are the endothelial progenitor cells. They are, this is a common marker actually for also hematopoietic stem cells and progenitors. And what we did here is a migration assay. So the mesenchymal stem cells in the scaffold are plated in the lower part of this well. This is a membrane that has very small pore, too small for the cells to go through passively. But if they are really being attracted by something that is secreted in this lower well, the cells will go through. Yeah? This is a transwell migration assay. And this is, you can see here that, of course, if you have the scaffold alone, very few endothelial cells want to go in there. If you have it with mesenchymal stem cells, lots go in. And now, if you specifically are blocking this signal that was being secreted by the mesenchymal stem cells, you again see less attraction of the cells, right? So we are basically saying we can activate the scaffold by adding these cells, and the reason why these cells help is because they are secreting this protein. This is, of course, one of many proteins that the cells are secreting that is important. Here are examples just of a couple more, right? All these are endothelial growth factors. They somehow affect the migration or proliferation of cells that form blood vessels. Yeah, vascular endothelial growth factor is very famous, and interleukin-8. I will come back to this VEGF, so that's why I'm showing to you. And the other reason why I like to show you this panel is because we think that a very positive effect on the culture of mesenchymal stem cells has been to put them in low oxygen, yeah, what we call hypoxia. So normal atmospheric oxygen is 20%. We like to put the cells in a lot less because they are not used to that level of oxygen in our bone marrow. We speculate that that's the main reason why they show so much positive effects when we put them in lower oxygen levels. The bone marrow is a place that has at the highest 5% oxygen and low to maybe 1%. So that's more or less the range where we are trying to culture the cells. And what we observe, among others, is, for example, that when you put them in low oxygen, they start to secrete more of some of these angiogenic factors. Yeah? For example, angiopoietin and this interleukin-8, others actually go down. So we need to be very careful if the overall picture is a positive effect or not. This is vascular endothelial growth factor, a very well-known factor to be regulated by hypoxia. And if we put them actually in 3% oxygen, as you can see, there is an increase of how much of that was secreted by the mesenchymal stem cells. But now the second questions come, well, okay, we have to define what's the best oxygen to give to the cells, but for how long to? Because if we culture the cells for just 2 days more, instead of only, they actually secrete even more vascular endothelial growth factor. So there will be, our task at this point has been to not characterize something that is well known in the field, that vascular endothelial growth factor is increased after exposure to low oxygen. This is about identifying what's the optimal concentration of oxygen and time that we have to give to the cells so that they're at their peak of recruiting endothelial cells at the time of fixing a wound. Yeah? For example, here again, when we culture the cells under these different concentrations of oxygen, I told you 20% is atmospheric, 10%, 1%. The less oxygen, the cells start to proliferate less. But that's only true after almost 5 days in culture. Before that, the cells didn't care if the oxygen was much. So there is also an effect, a chronic effect induced by hypoxia. It depends on how long you have been leaving the cells in hypoxia to start to see an effect. And this, for us, has been very promising because we think that lower proliferation means less metabolic demand, and those cells can be tougher. It sounds quite contradictory. It sounds like, well, the cells are growing less, they should be more unhappy. But actually, I think about it as the cells are used to an environment that is not the best for their growth in some sense, and that allows them to survive better when the situation gets actually much worse, which is when we put them in a wound. Of course, that wound, the naked scaffold, it's an extremely hostile environment. The mesenchymal stem cells normally there would die within a few weeks. And what we are trying to optimize is a method to make the cells stay there for longer. This is a very promising experiment we did in that regard. So all cells were cultured normally we culture them with just atmospheric oxygen. Now we wanted to study what happens when we put them in low oxygen. We either didn't do that for 6 days, or we cultured them only the last day, 1 day, with 1% oxygen, or we cultured them for 6 days with 1% oxygen. So this is a quite severe hypoxia. Yeah? And at that moment, what we did was to take off what we call the fetal bovine serum, which is like the food of the cells in culture. So in other words, of course, cells without food die eventually, right? It's a starvation process, and the cells have to undergo apoptosis and give up because there is no nutrients. So that's what we did. And we put them, of course, in very low oxygen, because that wound, as I showed you in the animal model, doesn't have any blood vessels. It's a very hypoxic environment, probably anoxic actually, so 0 oxygen, and makes the cells very difficult to live. So they are not having nutrients, and they're not having oxygen. All the consequences of not having the right blood vessels on the site. Well, we did this experiment. And this is what happens. We measured now apoptosis. Apoptosis is a mechanism that the cell uses to program its own death. The cells at some point have to say, when they are under starvation like this, after, in this case, 9 days without any fetal bovine serum, the cell has to decide to activate a cascade that will end up suiciding the cell. That's a process called apoptosis. And we can measure it by this annexin V5 method that I'm not going to go into detail now. But the important thing is that you can see that the cells that were in very happy conditions, in theory, proliferating the highest, when you put them immediately into this condition, you have almost a third of the cells dying. If you culture them for 1 day in oxygen, that was dramatically reduced. So this is you see that I'm cutting here. These are the still alive cells, and this black bar is now showing you the percentage of cells that are undergoing apoptosis, dying. This is at 33%. This is at 10%. If you have them cultured with 1% oxygen for even longer, it's even lower, also the percentage of apoptotic cells. So this is very promising to do now experiments in vivo, in animal models again, where we are putting the cells, having them cultured before in low oxygen, and hoping that the cells will retain in that tissue for longer. It's this type of the experiments. This is actually not the results we have obtained, because the experiments are really on their way right now. They're going to just finish like in the next 2 or 3 weeks. But they look exactly like this. These are mice where we are injecting the mesenchymal stem cells that have been genetically modified to express luciferase, yeah, from the fireflies, so they make light. And you can see here the presence of the cells, and as the days go, you can see that this decreases dramatically, right? Basically saying that the cells are dying. This is our method to measure how the cells are persisting in the tissue without having to sacrifice the animal. We actually can follow this experiment for as long as it's necessary. Yeah? In this case, we did it for 21 days. It goes here. And you can see that the cells are dramatically dying. This mimics very much what happened when we removed the fetal bovine serum in vitro. So that in this time frame, about half of the cells are actually dying, right? This is the original percentage of cell we injected, and they're coming down. This is a different study where we actually were looking for if the levels of sugar in the media could have a positive effect on the retention of the cells. And I think it's pretty clear from this graph that there is no real difference, at least in this setting of having the cells cultured for 1 week, in this case, before with 4.5 grams of glucose or 1 gram of glucose. Yeah? But we hope to obtain very different results if we are culturing now the cells with low oxygen or high oxygen. That's what we are currently doing. Yeah? So this is just to summarize what hypoxia can be doing for mesenchymal stem cells. It could be increasing, it actually increases the angiogenic potential of MSCs. We are still trying to optimize what is that time and concentration of oxygen that will be the ideal to obtain the highest levels of angiogenic factors. It affects the proliferation, and I didn't show you, but it actually inhibits completely the differentiation potential of the cells. So mesenchymal stem cells, when they are grown in very low oxygen, cannot become bone or fat cells anymore. And if we are planning on putting the cells in the scaffold of a skin, probably that's a very good idea. If the cells cannot differentiate into any other cell type and just will stay there as they are, that sounds very positive. Because we don't want to have bone in the skin or something like that, right? And as I just showed you before, the hypoxia are protecting the cells. Yeah, and this is a very important step because when we are thinking of this therapy where we are putting the cells in the scaffold, we ideally are putting that scaffold only once in the patient and not again every 2 weeks or something like that, right? So we need the cells to last there for quite some time. Another way to improve MSCs, make them better than what they are already, is by modifying them genetically. So what we did is to overexpress growth factors that are well-known proteins secreted by the cells that induce the growth of other cells. And it has actually, these specific growth factors are very well known to be important in the regeneration process. So, for example, the amphibians that I showed you at the beginning, a salamander, if you cut off the limb, it will show on that side extremely high levels of FGF2, right? It's extremely necessary in order to build that new build. And that's true for all these 4 growth factors. So that's why we took them all separately and overexpressed them. So we made the cells, modified the mesenchymal stem cells genetically so that they will make a lot of these growth factors. But what we did first is to characterize, well, what would happen to the MSC itself? There is an autocrine effect possibly, right? So maybe we are having some positive effect on enhancing the regenerative signal that the cells are secreting, but there might be also some detrimental effect that is affecting the MSCs in an autocrine level. And this is what this project was mostly focused. You don't need to, we don't need to go too much into detail, but this is a lentivirus. So it's the technique that we use to genetically modify the cells, to insert in them an extra copy of these growth factors, yeah? Basic fibroblast growth factor, platelet-derived growth factor beta, transforming growth factor beta 1, vascular endothelial growth factor A. And the important thing is that they are being secreted. Normally genes are secreted, are regulated, by promoters, and this is a promoter that is all the time on. So no matter what's the situation, the cell will be doing a lot of these growth factors. And that was our goal, right? To put them, for example, eventually on a wound or any other regenerative therapy we are developing with MSCs, and have them secrete, for as long as the cells are staying there, a lot of these regenerative signals. Well, first let's look at what happens to the cells. One thing that we observed is that overexpression of FGF and PDGF specifically enhances a lot the proliferation of the cells. And that has, of course, now sounds like danger. If the cells are growing too much, that's eventually a tumor, right? That's any cancer is characterized by that. It's a cell type that is proliferating indefinitely. And remember that I'm telling you that these growth factors are being secreted in a non-controlled manner. We are just making the cells, force the cells to make a lot of that signal. So from this already, I would say that even though these signals are, of course, very positive from a therapeutical point of view, the approach of engineering mesenchymal stem cells to overexpress them could have some consequences. Then we differentiate the cells. We took MSCs and added media that will induce the cells to become a different cell type. That's differentiation. In this case, osteoblasts, to become bone cells. And bone, of course, looks white, not red. But to make sure that it's bone what we are seeing, we stain it with something called alizarin red, which specifically binds to the calcium precipitates. So that's why it looks red. And the more red means more calcium. Yeah? Again, the 2 factors that were inducing a high proliferation of the cells are also forcing the cells to become a stronger bone than normally. In contrast, transforming growth factor beta 1 overexpressed reduces the effect, and VEGF almost no effect. I should have mentioned that also in the proliferation, the overexpression of VEGF really didn't have any effect on the cells. And this, for the sake of time, I'm just going to skip it, but it's another method to determine osteogenesis. This is about forming adipocytes. Now, we are taking the cells and adding a specific media that will induce the cells to differentiate towards fat cells. Yeah? Fat cells are characterized by this big lipid droplets. They're really full of fat. And we can stain them with a dye called oil red O. and probably the figures are too small. Here you can see them quite pretty. They look like grapes or so. And you can see that the overexpression of PDGF or TGF-beta completely reduced that, abrogated it in case of PDGF. There is some slight effect on FGF. It depends on really the method that you use to read it out. And again, really almost no effect by overexpression of VEGF. Transforming growth factor beta 1, as I showed you before, TGF beta 1 was completely blocking the potential of the cells to become bone cells or to become fat cells. And actually, we are not sure what the cells are becoming then, but they're definitely becoming something strange. They have a very distinct morphology characterized by very strong, what we call stress fibers. It's characteristic of a senescent cell, a cell that has become very, very old. And it shows high expression of certain markers. This is a smooth muscle marker, calponin 1, and this is a cartilage marker, collagen type 2. Yeah? And both of them are very highly expressed in the cells that are producing TGF-beta. So the cells are probably not becoming really a muscle cell or a true mature chondrocyte cartilage cell. It's rather an intermediate step that is just a, so to say, genetic effect of having make the cells produce so much of this growth factor. But once again, I want to characterize how the overexpression of VEGF pretty much didn't differ from the controls. So now we look, well, is it true that the expression of this growth factor has a positive effect? For this, we did a migration assay of endothelial cells. So we take endothelial cells. These are isolated, in this case, from umbilical cord of babies, and that would be normally discarded, thrown away. We take that cord and we take there is one vein in the umbilical cord. We take the cells that are making that wall of that cord. And we can isolate those cells and expand them. Those are called human umbilical vein endothelial cells, HUVECs. And when we add now the supernatant, so the media that was secreted by the MSCs that were, again, genetically modified to overexpress one of these growth factors, and we look now on how fast they are able to migrate. And the way we do it is by culturing a layer of these endothelial cells, and then we create a gap, which you can see here. There is here are endothelial cells on one side, on the other side, and then there is a big gap in between. Yeah? And you can see that when you add, for example, FGF, this gap is much, much smaller. With PDGF, maybe TGF-beta not really looks like this, and this, of course, is almost closed. We can quantify this, do this experiment many times with many endothelial cells from different donors, many mesenchymal stem cells, overexpressing these different growth factors. And we measure, after 12 hours, how much of this artificial wound has been closed, right? So the endothelial cells were able to migrate and close this wound. And it's clear to us that only the overexpression of FGF and VEGF actually had a positive effect on closing the wound. But again, I told you that the overexpression of FGF actually had many other effects that we actually don't like in the potential use for a clinical trial, because the cells are growing too much, the cells are becoming bone in a much easier way than they would otherwise, they are not able to become fat cells and so on. So the cells are pretty altered. But I had also showed you that the overexpression of VEGF actually didn't have any effects on the cells on an autocrine level, right? So now this is actually a very promising target. Oh, I think this slide. There you go. So we use an animal model again to study this. This is an hind limb ischemia. We take our mouse and we remove the femoral artery from their leg, from a posterior leg. And this is a technique called laser Doppler imaging to now measure the blood flow. And of course, if you remove the artery that goes to the leg, they have very, very poor blood flow. And you can see it as it's very blue, which is the background signal, right? This is the normal leg. This is the tail of the mouse. And when we inject mesenchymal stem cells, actually mesenchymal stem cells by themselves have already some positive effect on a regenerating flow. This is just the anatomical idea of this. Yeah? We are excising this artery. It's the most important blood vessels that bring blood to the foot. We're taking this out, and we are measuring how much of this signal comes back here over time. And this is something we did with the mesenchymal stem cells that were either modified with a control vector, doesn't signal to any growth factor, or with the VEGF, which was looking so promising. And as you can see here, it's clear that overexpression of VEGF actually has a much, much better effect on restoring blood flow to the leg as compared to the MSCs alone. And this actually has moved forward now. We are hopefully starting a clinical trial with this in the next few months in July, where we are treating now patients, really patients that, again, have typically diabetes, and they have this secondary disease that is called a peripheral artery disease. It's a very severe situation where the patients are not receiving any blood to their legs. At the beginning, it's just very mild, and you can notice it in patients with diabetes because they have to stop every 3 feet to rest because they're already tired of walking and so on. But eventually that gets bad and worse and worse and worse, and eventually the patient has to be actually amputated, because it's a so severe gangrene, the tissues die because they are not receiving blood. So that's the group of patients that we are targeting, not patients that have now just a little difficulty to walk, but actually are in a very critical step about the doctor is basically deciding, well, shall we now amputate or shall we try something else? Because there is no medication at that point that can help. So this is something we are doing now in collaboration with Dr. Laird, who is the chief of the vascular center in UC Davis. And right in the Institute for Regenerative Cures, where we do our research with little animals and things, next to it is what we call the Good Manufacturing Practice Facility, which is a facility that allows us to do this on a clinical grade level. So it's an extremely sterile environment. It allows us to make cells that are so pure that it will not get any contamination of any type. The same is the vector that will be overexpressing VEGF has to be produced in that facility so that we can ensure that there is no complication afterwards in the patient, and because of, for example, we are not injecting the pure MSCs, but for some reason they are contaminated, right? That would make it even worse than the patients are already. So that's the critical step, right? To develop a new vector that we will use to treat patients with critical limb ischemia. It's really peripheral artery disease, how it's called. They will be manufactured in the next door of our institution, and then treating patients that are actually like a block away from that. And we hope to treat 10 patients within the next 2 years or so. It's what we call a phase 1 clinical trial. That means that, first of all, we are really looking for safety, right? Because even though mesenchymal stem cells have been widely used in patients and they have already showed sufficient safety, now we are talking about genetically modified MSCs, right? We have modified them so that they can be even more powerful in terms of producing new blood vessels. And that, of course, confronts new challenges that we have to evaluate. And all what I've told you now with vascular endothelial growth factors has parallels in many other diseases. Yeah, these are all just collaborations we're doing with different groups. For example, there is another group within the group of Jan Nolta that works on Huntington's disease, and they are injecting mesenchymal stem cells into the brain of patients with Huntington's disease that are overexpressing brain-derived nerve factor. So it's of course a very different approach. Now it's not about forming blood vessels, it's about forming new neurons and not allowing at least the neurons that are there not to die, right? IGF-1 to treat ALS is a collaboration we have with Dr. Nanette Joyce. We overexpress the same vector with VEGF or this glia-derived nerve factor to treat obstetricular brachial plexus palsy. It's a defect on children when they are born. They come out like this, I think, and then they break. The thing is that they break a nerve on the neck, and that is also very complicated to treat. So they are now planning on injecting the mesenchymal stem cells that are secreting these growth factors, because although VEGF is well known to induce fast endothelial cell growth, right, angiogenesis, what I was showing you before, they have also a very positive effect on neuronal growth again. So that's why they want to use this growth factor too, to again build the connection that is missing in those children. And finally, we have a collaboration to try to treat spinal cord injury. It's a paralysis. The patients that have this disease can really not typically walk, are completely paralyzed. By putting mesenchymal stem cells that will be secreting these growth factors, also glial-derived nerve factors and the brain-derived nerve factor that we are using for Huntington's disease. So there are many applications. Really, we could try pretty much any growth factor. It's important to do first that screening to find out if the secretion of that growth factor will have an effect on the mesenchymal stem cells themselves. But if they pass those tests that prove some safety, and we can use animal models that show some efficacy, that is exactly what the FDA wants to see when we want to move forward to a clinical trial. Okay, so overall, I have shown you so far that mesenchymal stem cells could be improved. They have already shown great safety. There is nothing to improve on their safety point of view, right? We are actually dealing with not any of the modifications we do may risk to make the cells less safety than they already are. So that's a part that we don't want to change. But we definitely want to have higher efficacy for some of their treatments at least. One of them is to culture the cells with low oxygen, and the other one is by genetically modifying the cells. And now I'm going to switch rails to the second part, where I'm actually going to use the same type of genetic modification that we do in the cells, but to address very basic biology questions on the cells. And I hope to show you that at the end, both things are highly intertwined. And when you are using the same techniques to study something, even though you may not have very clear for what is at the very beginning, what will be the disease that we are trying to treat with this, you will see that eventually things come up that can make it very useful. This is about microRNAs. MicroRNAs are very small RNAs. That's why they're called microRNAs. 20 to 22 base pairs, so it's like a primer size. Extremely conserved, so there are microRNAs that are identical in worms and C. elegans to humans. Not a single base has changed since they warmed to us. So that tells us that probably they are good for something. And in the case of humans, there are more than 6,000 different microRNAs that have been described. A third of them are very conserved among all these species. Actually, even in plants, many of the microRNAs that are found in plants are extremely similar to what we can find in animals. And the main characteristic of microRNAs is that they are not coding for proteins, right? Normally, we think our genetic material is coded in DNA in the nucleus that gets transcribed into messenger RNA. That messenger RNA gets translated into proteins. That's the normal paradigm in cell biology. But that's not true for every RNA. So the DNA can transcribe into RNA, for example, a ribosomal RNA or transfer RNAs. There are RNAs that have other metabolic functions in the cell, different than to become a protein eventually. So they're not messengers. And this is a different category that was identified rather recently, and are these microRNAs, which main function is actually to control the expression of the translation of messenger RNAs into proteins. They repress the formation of proteins. Okay, they are coded in the genome. Of course, originally they are like DNA there. They get transcribed under a promoter, exactly like any messenger RNA would. That get transcribed. This is a very long fragment. This could be the size of a gene, 1,500 bases or so. But that get cleaved, that get cut by a complex that I will approach with a little bit more detail in a second. It get cut into this pre-microRNAs that look a lot like what we you call a short hairpins RNA, because it looks like a hairpin, and it's a very well-known technology that we use to turn off genes. And we actually didn't know that the cells use exactly that same technology in an endogenous level. So we were inventing, and we thought we were geniuses in the 70s when we thought about shRNAs, but actually a cell has always done this in the same way. So the shRNA gets processed by a complex including dicer and forming eventually this long chain that came out of the nucleus ends up being this very small fragment, yeah? 22 bases is extremely small, and that's probably why it was so difficult to identify these RNAs, right? Because they have been, it's very easy to miss them. They are just too small to be detected. A normal PCR technique, for example, would not allow to amplify them, because to do PCR, we use primers, and the primers are actually the size of the microRNA itself, so there's not much we could do. Well, the important thing is that once this microRNA gets formed, the mature microRNA, the small bases, they have two different mechanisms that lead to the same consequence. Stop making that protein. One of them is by a complete perfect match with a target messenger RNA. The messenger RNA, remember, is something that is also exported from the nucleus coming out and binds eventually in ribosomes to be translated into protein. It's on that level that the microRNA interferes, binds to the messenger RNA by complementarity of bases, and in this case leads completely to the cleavage of this messenger RNA. So it gets chopped into small pieces, no protein could be formed. If that match is not perfect, then typically there is what we call a translational repression. So it binds, it avoids the formation of the protein. This is the ribosome here, but without inducing the degradation of the messenger RNA. So those are the small, it's a difference that at this point sounds very small, because in the big picture, both are leading to the reduction of a certain protein. Okay, but as I mentioned before, I want to come back to this thing, because the question that we want to address is, well, do microRNAs matter in mesenchymal stem cells? Right? What do they do overall? And what we want to do for this is to turn off all the microRNAs in mesenchymal stem cells. It's a very general approach where we could think, well, most protease cells will die. Well, actually, they don't die, and that's what I want to show you. This is the complex shown in detail. When the prime microRNA, this rather long fragment, gets processed into the pre-microRNA. The pre-microRNA is the short hairpin that gets exported into the cytoplasm. And this is formed by two proteins, DROSHA and this DGCR8. DGCR8 is also known as PASHA in lower organisms. This is the protein that we turned off in the cells. We used lentiviruses to completely knock that protein. And if you don't have this protein, which is extremely specific to the formation of pre-microRNAs, you actually are reducing all pre-microRNAs in the cell. Yeah? So this is DGCR8. We are using two different lentiviruses. It's similar, the technology that I showed you, to overexpress growth factors, but now we use an approach where we're trying to silence the endogenous expression of a growth factor. Not growth factor, in this case, a protein, this DGCR8. Yeah? So it gets downregulated. We are using two different ones. I hope that doesn't confuse you. Eventually, everything that we show with one should also be true for the other. The only reason why we're doing this is to precisely confirm the specificity of our effects, and not that this was induced because something else. It's almost, you could call it like a duplicate of the experiment. But it's actually a lot more than that. And here we picked randomly 10 microRNAs that we know are very highly expressed in mesenchymal stem cells. And when their normal level is here in 1, this is the level when they are reduced. After having silenced the DGCR8 protein, this is now measuring the mature microRNA, the 20 bases. Yeah? And this is, you can see that they're typically downregulated to about half or so. That, as I mentioned before, has surprisingly the consequence, this reduction of microRNAs, no consequence on the survival of the cells. So the mesenchymal stem cells themselves don't undergo apoptosis with this. And I will come back to that in the future, because actually microRNAs are not present in all cell types and have a specific function once we became multicellular. I will come back to that later. The important thing is here, we're looking at proliferation, so how the cells can grow. And when we have reduced the microRNAs, also the growth of the cells has been strongly reduced. If we now induce the cells to become osteocytes, you can see that we are using pretty much the same assays that we used to study growth factors, now to study the overall basic biology questions of what are microRNAs doing in general in MSCs. This is the assay that we use to measure calcium, right? We stain with alizarin red the calcium precipitation. And you can see pretty much no red or no calcium after having turned off the microRNAs or downregulated the microRNAs. Yeah, this is the quantification of it. And this is, again, this other method, it's alkaline phosphatase activity. It's an enzyme that is highly expressed while you are becoming a bone cell, an osteoblast. And you can see that it's not so high when the microRNAs are turned off or down. Interestingly, when you look at this on the genes that are involved in the process of osteogenesis, most of them we didn't see any effect. So there is this core binding factor, alpha 1, it's a very well-known transcription factor involved in osteogenesis, and in fact, becoming bone. It seems to go down, but it didn't reach statistically significance, maybe to a half or so. No difference in this osteocalcin, bone yellow protein is robustly turned off. So the lack of micronase or the downregulation of micronase had a clearly very strong effect on the expression of bone yellow protein. That's also true on a protein level. So we can do a Western blot instead of a PCR, and instead of looking at the messenger RNA, we're looking at the proteins. This is a loading control. It just means that we everywhere put proteins, of course, but you can see that there is no BSP, bone yellow protein, if we have downregulated the micronase. When we look at adipocytes, this is a bigger picture to show you how this looks, the cells that are full of these lipid droplets. Yeah? And this can be quantified, in this case, by flow cytometry. And we also see here that normally you have around 15% of these cells in the whole layer would look like this. Now it's about 5%. And these are other markers. Again, we're looking at the gene expression of markers that are characteristic of that differentiation. Yeah? Proliferator, peroxisome activated receptor, gamma, PPR gamma, it's a well-known transcription factor, fatty acid binding protein 4, and adipsin, all of them go quite down after having downregulated the micronase. So with the shRNAs, that's what this nomenclature means, shRNA against the GCR8, 1 or 2. Yeah, that's the case for all of them. So overall, our idea is that low microRNA inhibit proliferation and the differentiation of the cells. Yeah? And it confirms the overall hypothesis that is now in the field that they are related to cell differentiation and developmental patterning. Actually, yeast cells, for example, which are unicellular eukaryotic cells, don't have microRNAs. At that level, you can perfectly live without microRNAs. The need of them becomes once we become pluricellular. Having many cells implies that they will have different functions, and that implies differentiation. And that's when microRNAs are starting to play a role. And that's, I think, also our understanding from these experiments. Now, the big question is, of course, what are the microRNAs involved in this? So we just looked at the big picture, but the mesenchymal stem cells are, of course, secreting quite a few of them, which is specifically involved in each of them. And some people have been identifying quite specifically, for example, microRNA 21 is important to become bone cells. Right? They can do that simple experiment. But it's quite biased in the sense that they are actually looking from the other way around. They're looking at taking mesenchymal stem cells, differentiating into osteoblasts, and looking for what microRNAs are differentially expressed from the beginning originated cells to what you ended up. If the microRNA was, for example, highly increased, it's a good hint that probably it's important during that process, right? So that has been a very common approach. We tried to look at this from a different one, and it was to really say, well, let's look at the function of the most abundantly expressed microRNAs in MSCs. Just whatever is mostly there. And so far, 5 groups have measured all the microRNAs that could be expressed in MSCs. They are using microRNA technology, so they have certain limitations, but they are looking at least for the first, let's say, 800 to 900 microRNAs described in MSCs. And out of those, they have a quite good consensus on 31 microRNAs. These 31 microRNAs, at least 4 of the 5 groups, the table is about 31 microRNAs that at least in 4 of the 5 publications were shown to be highly expressed in MSCs. And you can clearly see that the LET7 family of microRNAs is highly predominant here in this. So all microRNAs are typically just numbered from 1 to 6,000, but the LET7 microRNAs, which, by the way, were the very first ones to be discovered, form their own family because they are very similar to each other, as I will show you soon, and therefore they have their own name. This is what I wanted to show you. LET7 microRNAs, therefore, are very highly expressed in MSCs, are what's most highly expressed, and there are at least 7 different members of this family. The family in total in humans has 11 members, and MSCs are expressing at least 7 out of those 11 in a very high level. As I mentioned before, the LET7 are highly conserved. Flatworms, the single LET7 that they have is identical to our LET7A, the one we call LET7A because we had more. It stands for lethal because if you turn that LET7 off in microRNAs in worms, it actually kills this worm. So, and they are related in that, in those original studies in the year 2000, to progression in cell cycle and proliferation. They block differentiation in embryonic stem cells and are very highly expressed in differentiated cells. Mesenchymal stem cells, therefore, are closer in that sense to a differentiated cell, right? They're pretty far from what is the embryonic stage, where the LED7 levels have to be very low. And it's also important to mention that it's a known tumor suppressor. So if you have low LED7, it's actually a very bad sign overall for cancers. So it's been correlated with many different types of cancers, probably because it's precisely favoring cell cycle progression and proliferation. Yeah. So to have low of this doesn't allow the cells to move their normal path and get like stuck in one level, and that's typically of a tumor. Now, how to study LED7 and microRNAs, how to turn off one by one, sounds extremely challenging because the different members of the family are so extraordinarily similar to each other, right? So this is LED7A, B has only 2 bases different from A, and C only 1 different. So they have enough difference so that a single shRNA method to silence microRNAs, as we have turned off, for example, DGCR8 before, is not sufficient, will not work to turn them all off. And at the same time, they are different enough so that to turn off one actually will still have the others highly expressed there. So it becomes very difficult to, how are we gonna study the function of LED7 microRNAs if all of them, or if 7 of these members are being expressed where there's some overlap of function. They are similar enough so that they have some common target genes, but they also have differences so that some actually target genes are specific to a single member of the family and not to the others. Yeah? As I showed you, the MSCs are secreting these first 7, I think, or not G, but I, and so on. So what we did is to take advantage of a protein that was discovered, not surprisingly, in exactly the same publication where LED7 was firstly discovered, which is the first paper discovering microRNAs too. Yeah, it's a groundbreaking work in the year 2000. It's this Lin28, yeah? It stands for lineage protein 28, and it was shown, in that time the publication was, of course, in worms. It binds to LED7, and LED7 binds to Lin28. And it binds it in a way that it turns it off. So now, a decade later, we have found out that there are different mechanisms on how Lin28, this protein that is here as a cartoon, is able to repress the expression of LED7. It does it in the nucleus, stopping the pri to become pre-LED7. Remember that this is also the level where DGCR8 is actually a common protein for all microRNAs. Well, Lin28, in great contrast, specifically only targets LED7 microRNAs and doesn't touch the rest of the LED7 of the microRNAs. Something I will show you in a minute. And also in the cytoplasm, so even if the pre-microRNA made it out of the nucleus, the Lin28 will repress the formation of the mature form of this small 22 mature form of microRNAs. Yeah, either by repressing on the dicer level or inducing this pyruridination that also sends to degradation. No surprise. For example, embryonic stem cells that had very, very low levels of embryonic stem cells, of LED7, have very high levels of Lin28. And differentiated cells, like mesenchymal stem cells actually, that have high levels of LED7 don't have any expression of Lin28. So we modified the cells to make a lot of Lin28 in them. And that would be the way that we will turn off the LED7 members, the entire family. So this is a mesenchymal stem cells that are, again, transduced. We are using lentiviruses for all these genetic modifications. And you see that the protein that wasn't there suddenly is there. That represses LED7 as expected. These are the same 10 microRNAs that we had examined for DGCR8. DGCR8 was bringing them all down to around half, right? Now only the LED7 members that we screened are downregulated, and the other microRNAs stay very close to their basal level. Am I going long? Yeah. So, and This is a very well-known target of LED7 microRNAs. So it's a gene that typically is repressed by LED7. If we have now turned off the LED7, no surprise that target gene actually goes up, right? We are turning off the repressor, so it's increased. No effect on proliferation. This is in great contrast to what happened with the total silencing of microRNAs. It's saying that the LED7s are not responsible, at least for that part. In osteogenesis, this is adipogenesis, forming adipocytes. There is some effect, but it's milder than what we saw compared to what the silencing of all microRNAs does. And on bone level, the effect is also rather mild. It is, however, extremely similar on the bone scalloprotein, right? Remember that the other markers, osteocalcin and CBFA1, were not changed? Well, the same is true here. Alkaline phosphatase was also strongly downregulated, and this bone scalloprotein was completely abrogated. Also on a protein level that I don't have to show you here. And these are just the summary of what they have in common and what they have different, turning off all microRNAs or having turned off only the LED7 members. Yeah? Proliferation went down. The responsible for that are not LED7 members. It must be something else. There is no induction of apoptosis. There is a strong reduction of adipogenesis, which is milder when you turn off only LED7. The markers, however, are very similar. Osteogenesis is also milder, and very similar as the effect on bone scalloprotein. And this is the final slide. I just want to show you what we are currently working on, where actually these different projects are kind of coming together, right? So from one side is, of course, to continue optimizing the MSCs for the scaffolds in wound repair, the precondition of hypoxia and possibly genetic modification if that brings any advantage. Now we're looking, for example, for what microRNAs are differentially expressed after exposure to hypoxia. This is something that we don't know, and this could have consequences because maybe we could, instead of putting the cells in a hypoxia, modify specifically that microRNA that has been responsible to, for example, enhance the angiogenic signals or promote the longer retention of the cells in the body. And this is something that I didn't show you sufficient data to show you how actually LIN28 has also some positive effect on mesenchymal stem cells, and this is something we want to explore to improve actually osteogenesis with them. And this is where I want to end. I just want to thank especially my mentor, Jan Nolta in the lab, and the people I work with very closely, Stefanos Kalomiris, Klaus Sondergaard, Heather Stewart, Julie Beagle, and Elizabeth Clark, and there's a lot of more people in the lab. Our collaborators are Jana Bad, Kent Leach, Thomas Peavy here, and Rivka Isurov. And this has been funded by itself. Thank you.